It is with great excitement that I'd like to announce our keynote speaker to start off the symposium, Tim Jennings. Tim is a graduate of Longwood's Professional Gardener Training Program and is the senior gardener responsible for Longwood's outdoor display. He teaches several popular water gardening courses at Longwood Gardens and also serves as lab instructor for various plant identification classes. Tim helps the IWGS with the Victoria Conservancy Program and the new water lily competition. You are being treated to a presentation on Victoria, the giant water platters at Longwood Gardens. I am very excited and pleased to introduce Tim Jennings.
Well, welcome everybody, and thank you, Justin, not just for the introduction, but for remembering those that we all have met and been influenced by in the past. And you are right, it is up to us to kind of carry on their legacy, if you will. So today, as part of that kind of carrying on of a legacy, I am very excited to continue our discussion on the giant water platters here at Longwood Gardens. But before we get started, I kind of thought I'd like to show you a short 4-minute kind of overview of the pools at Longwood, especially for those who may not have had a chance to visit Longwood yet, to see kind of what Longwood is all about as far as the water gardens. And so enjoy this little 4-minute clip, and then we'll see you back to get started on the presentation.
Well, good morning, everybody. Welcome to Longwood Gardens. My name is Tim Jennings, and we're out here in the water lily display at Longwood Gardens. You'll notice some noise in the background, and I wanted to make sure you knew that we are in the midst of one of our largest renovation projects called Longwood Reimagined. Today, we're standing in the center pool with the giant water platters, which will be the main feature of our talk today. These majestic giants are probably one of the most iconic pieces of our summer display. Besides the Victoria water platters out here, we have well over 100 different varieties of water lilies. Here is one of my favorite. This is the tropical day-flowering water lily known as foxfire. You'll notice the flowers are beautiful and the leaves are absolutely exquisite. So another of our tropical water lilies that's a favorite here at the gardens is this night-blooming water lily called Trudy Slocum. What a magnificent plant with its big, big white flowers. It almost doesn't need any light at all to be seen at night. So here we are in our hardy section of the water lily pools, and we're looking at one of our newer water lilies, Siam Rose, which looks particularly beautiful this morning. Wow, what a beautiful plant. This is another one of our new plants here at the gardens. This is called Detective Erica. Detective Erica is different, and it is actually a hybrid between our tropical water lilies and our hardy water lilies. What an amazing plant. It's proving to be fully hardy for us and an absolute find. So we're in the pool today with the Santa Cruz water platter. This is Victoria cruziana. This is one of the parents to the Longwood hybrid water platter. Take a look at this leaf. It's unbelievable. Look how high the rim forms on it. Look at the actual size of it. The plant just begs to be looked at. This plant has amazing, amazing attributes, not to mention the lip, not to mention its size, but here's a new leaf coming up, all ready to uncurl and expand at a rate of 6 or 7 inches a day. These plants will reach a size of 4, 5, even 6 feet in a matter of weeks. This plant has amazing, amazing attributes to it. So in this pool, each one of these platters is actually one leaf. Now let's take a look. There's even more to see on this plant. This plant has, besides the lip and its size, it is armed with one of the most vicious thorns of any plant I know. These thorns are as sharp as rose thorns, and they have a couple of purposes. One is they prevent predation from other animals, particularly the manatees, which live in some of the areas where these plants are. The other thing that happens with these plants is because of their size, these thorns will actually clear out areas as the plant grows, enabling them to expand. So we are in the pool here with the Royal water platter. This is Victoria amazonica. It is one of the parents to the Longwood hybrid water platter. It also exhibits that kind of red pigmentation that you'll see in the hybrid, as well as the large leaves and the upturned margin. We also have a first night flower on this one. This is the flower that opened last night for the first night. It will close back up and reopen tomorrow night, shedding pollen, which will be used to make the hybrid. Well, I hope you enjoyed this introduction to the water lily pools here at Longwood Gardens. If your travels ever bring you this way, please visit, because there's nothing like seeing these plants in person.
Okay, everybody. I think we're back online here. Welcome back. I hope you enjoyed that quick overview. It's very interesting. Each morning I go out, I could have picked 4 or 5 different plants to look at. It just is a very spontaneous thing for me to walk around in the garden in the morning and say, oh, this is my favorite plant today, or this is my favorite plant tomorrow. But each time I go out there, something new pops up and just begs to be looked at. So I know I sound a little bit like an infomercial in some of those pieces, but that's just me and my excitement for these plants. So as we go along here, we're going to go ahead and get started with the process of actually talking about the pollination of Victoria. But before we actually get there, I wanted to do a quick review of an earlier presentation we did for the Society, where we took an in-depth look at the seed and the sowing of the seed and the transplanting to bring everybody up to speed on this kind of cycle from beginning to end on Victoria. Then we will slow down a little bit and we'll actually talk about the pollination process as a whole from beginning to end. And then at the very end, we put together a video which will serve as a resource for you if any of you would like to dabble in the pollinating of these plants. So let's go ahead and get started with this presentation. And that's me, Tim Jennings. So it all starts with the seed. And what I found interesting is that you can actually tell at least one of these plants just by the seed. And if you take a close look at these plants, Victoria cruziana on the left, the hybrid in the upper middle, and then Amazonica on the right, you will see that Amazonica has a distinctly egg or oval shaped cast to the seed, whereas cruziana is almost entirely circular and round. And that trait is also carried through in the hybrid. So if you were to show a seed, I might be able to tell you if it was Amazonica, but I certainly couldn't tell you the difference between cruziana and the hybrid. These are just a few little things that we kind of picked up over the years as we went along. So if you have oval seed, you know for sure that you have Amazonica. If you have round seed, you could have either the hybrid or cruziana. Now, when we sow the seeds, we usually do our seed sowing in early February. We sow them in what is called a community pot. Here, the seeds are being sown 5 to a pot. That pot's about 4.5 inches in diameter. And we'll sow those seeds like you would any other seed. The seeds would actually get covered at this point so that they're buried in the soil. It doesn't take long, maybe 2 weeks, maybe 3 weeks. You'll start to see them first germinate. And if you're unfamiliar with what they look like when they germinate, you might think you have some weeds growing in your pot, because the very first seed that comes out is a very long, kind of grassy, filamentous type leaf. That's quickly followed by this deltoidy shaped leaf that you see in the picture there. And those 2 leaves make up what I would kind of call the cotyledons of the seedling. And it's not until you get to that 3rd leaf that you actually start to see something that resembles the parents. Here they are, that community pot, or a different community pot actually, grown up, and those seedlings with those leaves that are in the hand there are actually floating leaves, and you can see down below those deltoidy shaped leaves and remnants of that filamentous leaf. So what do you do with them from this stage? Here you have a pot full of seedlings, and these things must be hard to grow because, they're aquatic, and it's kind of this mystery that nobody really knows about. Well, step back a little bit. Take a look at them and just think, what would you do if this were a pot of tomato seedlings? Well, you're going to start taking them apart. It's called pricking apart. You take that pot, you put it in water, you soak it apart, the seedlings will start to come apart, and then you transplant them individually. That's what's happening on the right here. Once back in the water, it doesn't take long for them to get started. So here we are in mid-March. Maybe another week or a week and a half later, you can see the leaves are starting to double almost every week at this point. Here we are in April. April, we're moving right along. You know, those leaves might be 7 or 8 inches long or in diameter. Down on the right, by May, they are really starting to kick. The days are getting longer, the light intensity is getting brighter. We are starting to really see some good growth on these by even early May. We're starting to get leaves that are probably 15 inches in diameter. And then we get to the real time of the year when we're starting to get to the point where we're almost ready to put them out, which for us is the end of May, right around Memorial Day. And our bigger problem at this point is, how do I control the size of these plants? You know, how do I keep them from running over each other? How do I keep them from climbing out of the pots? So it's a better problem to have than to not have any plants, but it is still an issue at that time of year. We're constantly shifting plants around. You know, Justin's discussion on those that we've lost certainly brings me back to Patrick here. And Patrick and Ken had a great relationship, and it was because of Patrick that I got to meet Ken Landon. So here he is. He's retired at this point, but still gets a kick out of coming in and seeing these plants right before they go out. He just, a smile on his face, it could bring excitement to anybody. So this is Patrick. He was certainly a mentor of mine, and what more can you say about a man who dedicated his life to this field? So let's step back for just a second and take a look at the two parents and kind of compare them with some of the traits that each one has. On the left-hand side, bulleted items for Victoria cruziana. On the right-hand side, Victoria amazonica. So as far as nativity goes, our cruziana that we grow here at Longwood are primarily from the Paraguayan strains that came from the Missouri Botanic Garden many, many years ago. But they also can be found in Bolivia and Argentina. And these are cooler areas to grow. I mean, we've found seeds and plants growing of Victoria cruziana in Argentina later in their season, and the water temperatures were down in the 40s. It was amazing how cool the water was. Contrast that to Victoria amazonica, and you have Guyana and the Amazon basins of Brazil, where these plants primarily grow. Again, we've already mentioned, cruziana seeds are perfectly round for the most part, and amazonica seeds are actually oval and very, very distinct to tell them apart. You typically, with Victoria cruziana, get a much higher upturned leaf margin, whereas the amazonica, as you'll see in a little bit, ours have maybe an inch at the most of upturned margin. When you compare that to cruziana, we can get 7, 8 inch high lips turned up on them. Some of this upturned margins might have to do with light. I know they were finding very high upturned margins in some of their trips to the Amazon regions of Brazil and some of their collecting trips. But when we grew them out in this climate, in this kind of temperate climate that we're in here, our margins never got more than about an inch high. The leaf color, that light green color, I mean, to me, Victoria cruziana is an amazing plant to try, especially in the home garden. If you're a home gardener and want to try a Victoria to grow at home, either Victoria cruziana or the hybrid would certainly be better choices than amazonica, because amazonica can be a little finicky to get started, and it can be a little bit more demanding in its water temperature requirements. Also on the amazonica side, when you compare the leaf color, that red pigment that comes through, especially in the lip when it's backlit by the sunlight, really, really shows up well. Now cruziana's got a flower, and we're going to talk a little bit about the flowers here later, but the Victoria flowers flower for 2 nights. That first night, they're both white, but on the second night, the cruziana tends to be definitely a lighter pink color than the amazonica, which can be almost burgundy in color on the second night. So these are just some very visual characteristics that you can use to help distinguish between the two. And here's kind of another slide, very similar, but down below are some pictures kind of highlighting some of the things that we talked about. One of the things that wasn't on that first slide, if you take a look at the flower on the Amazonica and the flower on the Cruziana. And if you look closely, you'll notice that on the Amazonica, the sepals that protect that flower bud are actually covered in thorns. And then if you look at Cruziana, one of the characteristics of Cruziana is that there are few, if any, of these thorns that kind of coat the sepals that protect that flower. And when I say thorns, I'm looking down at my list here. We actually sometimes interchange terms, and it can get a little confusing. But when you look at what we call thorns on Victorias, if you look at the anatomy of them, they're actually called prickles. So they're not thorns at all. They're just another term for them is prickles. So to create the Longwood hybrid, there's a specific way in which you make that cross. So if you look at Victoria Amazonica, it is always the pollen parent, meaning that it's the one that bears the male characteristics of the plant, versus the female parent, which is always going to be Victoria cruziana, which means that basically cruziana is always the receptive parent, or the one that's going to have the pollen spread on it, where it would then germinate and then unite with the eggs to form the seeds. So here's a picture. In the foreground, we have Victoria cruziana, high upturned margins, lighter green color. In the background, you'll notice that's our Victoria Amazonica one year, and you can barely tell that a lip is starting to form on it. And there's a shot from the other angle showing you Amazonica, and just the slightest lip forms. This year, our lip is about 1 inch high. So one of the things you want to remember when you're growing Victoria is that since these are seed grown, as with any other plant that's seed grown, you're going to get some variability from year to year, which makes each year unique and actually fun to grow. So this year, very little bit of a lip was formed on the Amazonica, but some years we'll get a lip that might be up to 1 inch or so high. The same with cruziana. Some years those lips are exaggerated, and some years they are maybe only 3 or 4 inches high. And there was some earlier discussion just this week about, some of the things that might be controlling that. And we're going to really start to take a look at some of the environmental conditions that might influence what the plant's potential has as far as growing leaf size and growing the lip extension as well. So there's a lot more to come with research on Victoria. So you take those two plants and you cross-pollinate them, which Patrick did back in 1959. He made the first cross, and this is what you get. This is the Victoria Longwood hybrid. So it takes many of the characteristics from the female side, or dominant, but it does pick up the red pigment from Amazonica, especially in the lip. At certain times of the day when the light is low and shining through, it almost glows out there with the lip shining in this kind of red pigment that's picked up from Victoria Amazonica. The other thing that happens just in the natural process of creating hybrids is you get what's called hybrid vigor. So these plants oftentimes outgrow the parents, as well as producing more flowers. So instead of maybe getting 10 or 12 flowers in a season, 15 flowers, we might get 30 flowers total on a hybrid water platter flower. So more flowers and more vigor are typically things you'd see from creating a hybrid. Okay, so that brings you up to speed to where we kind of left off with the seed sowing and transplanting demonstration we did earlier. And I believe that is also going to be available on the website somewhere, so you could, if you haven't seen that, you can start with that piece and then follow up with this piece, which again is going to be available to you all for some time, especially the members. So Victoria pollination, the process of hand pollinating. Let's take a look at what we have here. So because we are hand pollinating them, I started just take a quick look at the tools that we use. There's nothing special here. If you've done any pollinating at all, you probably recognize these tools and may have even used some of them. So we have scissors, we have a paintbrush, we have tweezers, we have a vial there in the middle that we use to collect the pollen. That bottle in the back has alcohol in it so that we can sterilize our tools, disinfect our tools in between plants if we happen to be doing more than one type of cross in a day. The yellow tag there is very important, and we'll talk a little bit more about that as we go along. And then down in the kind of the middle bottom here, the white cloth material is actually a polyester material. If you've ever sleeved plants like poinsettias or other hydrangeas, oftentimes plants will come in kind of either paper or these kind of polyester sleeves. The polyester sleeves we found are ideal for covering the flowers when we're doing the pollinating. This prevents them from getting any contamination, and it also helps later in the season so that when the seeds actually burst, you don't get the seeds floating all over the place. You can collect the seeds that you've had. So we're going to take a look at how these are used throughout the whole video as we continue. So in nature, this is what pollinates them. So this is one of the collecting trips made back in the late 1990s. They were able to bring some of the actual beetles back and get them identified. There are two beetles. This is the larger of the two beetles that are known to pollinate the Victoria amazonica in Brazil. So these guys are called scarab beetles. They're basically night flying insects that are beetles that actually can be anywhere from 1 to 20 or 30 beetles found in a flower down the Amazon when they were actually collecting seed samples and came across these beetles. So let's look. Up in the upper left-hand corner, a first night flower. Up in the bottom right-hand corner, a second night flower. You know, one of the things that's, that amazes everybody who hears about this plant for the first time is this transformation that takes place between a first night flower and a second night flower. On the first night flower of these plants, it is almost pure white. But if you look to the center, you can see that there's already a little transformation taking place as the plant prepares itself to go from a first night flower, which is predominantly female, meaning that it is receptive, to a second night flower where everything is kind of reflects back and the plant starts to shed pollen, which gives this plant a dominantly male characteristic. Actually beautiful flowers. The top one is actually Victoria amazonica, and so is the bottom one. So this is an amazonica flower on the first night, and that very burgundy, almost kind of burgundy colored flower on the second night. Absolutely stunning to look at. So we take a couple flowers and we start to kind of take a closer look at what's actually involved with these flowers. They are very, very primitive flowers, very simple in their structure. Let's just take a look. We've taken a cross section through this. So at the very bottom, we'll start at the bottom and work our way up. The bottom is the ovary. So the eggs that you see in the little yellow round things at the bottom are eggs that are ready to be fertilized. Characteristic of all Nymphaceae, all plants in the water lily family, is that stigmatic bowl, which you should be very familiar with if you grow water lilies. The same follows true with Victoria. That stigmatic surface, though, is a little different than that of water lilies, because when you open up a Victoria flower on the first night, it should be bone dry and it should almost be chalky on the inside. Sometimes when you open up a flower, you're going to find a liquid in there. Now, whether that liquid is a nectar or whether it is from water, I don't really know. But I do know that when you open up a Victoria flower, different from when you open up a water lily flower on the first night, you should not find the stigmatic fluid in there of any kind. It should be actually bone dry. Right in the middle, there's a little protrusion that's the only thing I've ever read about this is that it's a floral node. You can think of it maybe as the core to this flower. You can see how it almost kind of is the core piece to the actual ovary itself. And if you were to kind of peel, you could almost see how you could peel away the outside edges. The purpose of that core is, I still don't know if anybody out there has any specific information about the core. We'd love to hear from you as we're trying to kind of decide what some of these functions are for some of these pieces on this plant. So follow that up, and you're going to come to a spongy kind of material that's up there. I kind of almost think they look like piano keys. These are actually starchy appendages that serve as a food source for the beetle. So as the beetles enter this flower, they're going to find that this food source in there that kind of helps keep them in the flower until it closes back up. If you follow it up even further, you're going to come to these red kind of swooping structures. Those are anthers. These are what are going to shed the pollen. Oftentimes on either side of the anthers there, you'll get what are called false anthers or pseudo anthers. They're kind of immature anthers that are kind of sandwiched in between the actual anthers, and maybe their role is to help protect the anthers from the beetles eating at them or from the weather beating up the pollen. But we oftentimes find that on the very bottom and on the very top, those structures haven't fully developed into mature anthers. So there's this layer on top and layer on the bottom, which helps sandwich in between the anthers there. And then you follow it around, and then you get to the petals, and then finally back out to the sepals, which actually help protect the flower before it opens. So this is just a really quick look at the anatomy of the flower. It'll be useful as we start to look at pollination and what we're doing with the various pieces. Okay, so let's say we're ready to start pollinating. What do we do? So for us, we're always looking at what flowers do we want to cover. So later in the days after 2:30 or 3 o'clock, you can oftentimes go out to the pool and you can kind of start to tell which flowers might open for the first night. That will be the picture on the left-hand side. You can see the next morning, I would have covered this the day before this picture was taken. The next morning, you can actually see that the flower has popped open and that the flower is white. If we leave that flower covered for one more night, 24 hours later, go to the right, and you can actually look into the cloth and you can see that the flower has actually changed to that pinky red color. Okay, so this is the sequence that you're looking for when you're starting to do pollinations. You want to cover a flower before it opens because you don't want the flower to open and have a chance of getting contaminated. If you need to collect pollen, you'll leave that flower covered for 24 hours, come back in the next day, and then you could harvest pollen from the plant on the right. The pink one's going to be shedding pollen, the white one is receptive. And if you haven't picked up on my abbreviations, FN means first night flower. Sorry about that. Okay, so there it is. We open up the red flower and look how profuse the pollen is. This plant is not shy about shedding its pollen, that's for sure. It covers everything it touches. It touches the tweezers, the paintbrushes. So what we want to do, if you're ready to collect pollen, you'll literally go in here with scissors and actually cut the anthers. And then we'll take the anthers, the entire anthers. We're not going to collect the pollen from the anthers. We're going to take the entire anther and we're going to put it into a vial where we can then either use it or store it if we need to. So here's a second night flower shedding pollen. So in this case, we have two different vials here. The one on the right is Victoria cruziana. The one on the left is Victoria amazonica. Let's just say that we have flowers that are shedding pollen, but we don't have a first night flower ready. We found that you can actually hold pollen for up to 48 hours in the refrigerator. So that's 42 degrees in your refrigerator. You can take the pollen vials, store them in the refrigerator. I typically put them in where we store the butter, that flip top. It kind of keeps it away from all the other food in the refrigerator. You can store it there for 24 to 48 hours, wait for another flower to come open, and then use that pollen. Now, having said that, don't be, if you happen to store pollen and the next day you come in and you have fresh pollen that you can use, it's always better to use fresh pollen. So sometimes I'll store pollen and then I'll have another flower that actually will open. I'll just go ahead and get rid of the old pollen because the older the pollen, the less viable it becomes. So use fresh pollen if you can, but if you need to store it, know that you can store it and use it for up to 48 hours later. So now that we've collected the pollen, so you have the pollen in the vial, you either had it in the refrigerator or you're taking it to another flower, here's the next step. Now, I still had that first night flower covered. What I want to do is I want to uncover that flower and begin the process of figuring out what I'm going to do with the pollen. So if I have the same species, either Victoria cruziana or Victoria amazonica, I would simply cross the plant with the pollen from it. So if I'm going to create the hybrid, though, we have to go through a process of kind of removing any of the male flowers or male parts of the flower by a process known as emasculating. So here I've uncovered it. Take a look at something with this white flower, the first night flower on the right. You'll notice that on the first night flowers that the anthers are kind of standing up in almost an erect position, creating a cavity or a hole, if you will, that the beetle will actually enter through. The beetle actually climbs down into that flower through that entrance hole. Now, the other unique thing about this flower is that it actually heats up. It's 10 to 15 degrees warmer inside that flower than the outside air temperature. And if you're ever out there and you have the opportunity, stick your finger down in that hole. You'll be amazed at how warm that cavity is. That warmth that that flower generates is used to attract the beetle to the flower and may also play a role in helping the pollen grains germinate once the beetles move from a first night to a second night flower. So some of the beetles that are entering this first night flower are going to be covered in pollen. Those beetles will be ones pollinating the flowers in there. When that flower closes and reopens, it's going to be shedding pollen. So any beetle that leaves that flower will be carrying pollen from this first night flower, or second night flower in that case, back to a first night flower. So this will become a little more obvious as we move along. Just unbelievable to look at, though. It's just amazing, these flowers. So if we want to create the hybrid water platter, or even if we're doing the parents, I often go through this process that we talked about earlier called emasculating the flower. And what it involves going in to the flower and removing all of the anthers. Now, the anthers are in what are known as a ring, and if you follow the flower around the curve of the flower, it will actually come around and kind of tuck its way under right where those starchy appendages are. And you can take scissors and literally poke in there and cut through those starchy appendages and remove that entire ring of anthers to ensure that you don't have any anthers that would shed pollen that next night. So in this case, the flower's been emasculated and we have dropped in Victoria amazonica anthers, which we will be spreading around with a paintbrush. There we are. So here we are in that flower. We're actually using a paintbrush to emulate the kind of beetle feet, if you will, and kind of walking around on the bottom of that flower with the bristles of this paintbrush to spread that pollen evenly along the entire surface of this kind of stigmatic bowl. Okay. When we first started doing these pollinations 20 years, 30 years ago for me, we used to think that you wanted to be very delicate. You didn't want to disturb this area very much because you're afraid you might kind of damage that stigmatic surface. Then I met a gentleman named Billy Anderson, who some of you may know, down in Georgia. I went down and did some pollinating with him, and he found out that it actually could be beneficial to be a little rougher with that than you wanted to be. Don't be worried about, being real delicate when you're walking around that flower. Take that paintbrush and really make sure you kind of brush in that pollen so that it adheres to that stigmatic surface, because that's the first step. You got to get the pollen to adhere to the stigmatic surface where it can then germinate, send down a pollen tube, and then it would unite with the egg. Once the flowers have been pollinated, we leave the sepals on the flower. We'll literally take the sepals and we will use them to recover the flower. So we'll take those 4 sepals and we'll kind of crisscross them back and we'll tie them together so that it has a chance to protect that stigmatic surface, because it takes about maybe 4 or 6 hours for the pollen grains to actually germinate and unite, or germinate and then send down its pollen tube to unite with the eggs. So at this stage, we're tying it back together to prevent any rains that might come by from getting in there, and it makes it a lot easier when you go back to try to recover this, because now that we've pollinated it, we want to make sure that we recover the flower, because we don't want anything accidentally getting in there and causing any contamination. We went through enough trouble to make sure it was covered from the beginning. Make sure that you recover it at the end as well. So there we are again, we're recovering the flower. Remember, we're covering a first night flower, so you still see the white petal in there in the middle. We found that these floats are very useful in kind of holding the flower out of the water, because when you do these pollinations, you have to do them kind of with the flower still attached to the plant. And so sometimes you would push on the flower and it'll go underwater. So these floats aid in allowing you to do all of the things that you have to do to the flower, cutting off of the anthers, the rubbing of the pollen in the stigmatic surface, and so forth. So these are just 1/2 inch, 1 inch styrofoam floats that can be cut with a slot that would slide right in where the pedestal to that flower is. Once the pollination is done, then we want to make sure we tag it, because we're creating 3 different types of seeds. We're creating seeds of Victoria amazonica, seeds of Victoria cruziana, and seeds of the Longwood hybrid. And as we talked about earlier, the only one that you can really tell what you have is Victoria amazonica because of the seed shape. So it's very important at this point that you tag what you did for a couple of reasons. One, you need to know when you did it, because that'll give you a rough idea of when the seeds will be ripening. It takes about 6 weeks, 5 to 6 weeks, from the time you do pollinations until the time the seed pods burst. So if you do your seed pods on the 27th of, in this case, of August, it's going to be somewhere around the beginning of October when those seeds are ready to be harvested. Once the pollination is done, and you've tagged it and bagged it, it'll take about a week or 2, and you should start to see that ovary swell. It'll swell to be about the size of a small grapefruit, maybe even a cantaloupe. And in that pollination, if it was successful, you can have as many as 300 or 400 plus seeds in one pollination. So there's a lot of opportunity for seeds in each time you do a pollination. The other thing to tell you about these tags before we move on is that typically when you see something written out, a hybridization written out, you usually start with the female parent first. And so that's, in this case, it's Victoria amazonica from our bee pool. And that was crossed back in 2007. That's when we did this pollination with our Victoria amazonica that came from seed from Guyana. So that's what the G in this case stands for. Also on this tag, I would make a note of anything that was of interest or that I might want to know later. So the word fresh that you see written there means that we used fresh pollen. It wasn't pollen that was off of a ring that we had harvested. It wasn't pollen that we had stored in the refrigerator. It was a cross that was made by collecting pollen and taking it directly to the other plant, in this case, Victoria amazonica, which was in our bee pool. The other thing, that particular night, it must have been very warm and humid. So I just made a note that it was warm and humid. If there was rain around, I certainly would want to make note of that. So anything that you think that might be useful later on, just jot those notes down on this tag. And that tag will stay with the seeds all the way through until you start to sow the seeds in the spring. Now, in 6 weeks, when it bursts open, this is what you're going to get. So this is Victoria, probably a cruziana, I'm going to guess, seed. And you'll notice that it's just kind of, each seed seems like it's kind of sitting in its own little kind of mucousy membrane. And that is very intentional. This is the plant's method for distribution. So when that seed first bursts open, all of those seeds are individually wrapped in their own mucus sac, which enables the plant to float. And sometimes they'll call that an aril. These seeds will float. They can get up in the current. They can be eaten by the fish. They can be picked up by the birds. And they may float miles down the river. And this is very important for the plant. It knows it needs to do this to survive, because maybe next year, the area where this plant was growing, maybe that area dries up, or maybe there's an avalanche that fills this area in. Who knows? There's so many different things that can happen. Maybe somebody builds a parking lot on that spot. So you need to know that, the plants have in their already built into their memory ways to survive, and this is one of them. So they disperse their seeds as widely as possible by floating down the river or being picked up by birds or fish. So taking that from our cue, oops, I'm sorry, let's go back. So there they are, another close-up. You can see the little dots are actually eggs that were never fertilized. and mixed in there were all of the other seeds floating in that mucousy sac. So what do we do with those seeds? Once we collect the seeds, we take it as is, with just like you're seeing in this picture, and we'll literally float those seeds in water for about a week or a week and a half and let that mucousy membrane decompose naturally. We'll go in and rinse them off each day, and let that just kind of naturally happen. Let the process of decomposition of that kind of aril naturally happen, maybe 2 weeks or so, and you'll end up with the seeds will all sink to the bottom. The ones that float, you can actually get rid of because those are the ones that are dead. So all the seeds that sink during this process, are likely to be viable. All the ones that are floating, you can just go ahead and scoop them off and get them out of your way. So once the seeds, the mucousy membrane is decomposed, what happens in nature next? Well, wherever those seeds are, they're going to fall into the mud, into the banks or the rivers, and they're going to sit in this kind of semi-wet condition until the rivers rise again. And as the rivers rise, they're going to germinate and grow along with that. So for us, what we do is once the seeds have sunk to the bottom of our pot and we've removed all the ones that are floating, we take and we put these seeds in layers of moist sand. And this, you'll see the lighter color seeds and the darker colored seeds, as these things sit in these kind of layers of moist sand covered up for about, maybe 4 weeks, they will gradually go from being kind of a lighter green color to almost a chocolate brown color by the time they're done. So something happens in this post-harvesting stage with these seeds that allow them to kind of get themselves ready to stay dormant until the next time they're needed for germination, which is when the conditions are right again. And that, in our case, would be when we sow the seeds in February. So these seeds are perfectly fine to sit in this kind of dormant state right through the winter. In fact, we store all of our seeds, the Amazonica, the Cruziana, and the hybrid, in a cooler at about 55 to 60 degrees. What we found is that if it's stored much cooler than that, Victoria Amazonica has really poor germination, which it already has poor germination, but we rarely get anything to germinate if temperatures fall below about 55. And much above 55 degrees, and the hybrid and Cruziana start to germinate. If you remember earlier, I mentioned that they found seeds and plants of Victoria Cruziana growing in Argentina and water temperatures that were down in the 40s. So these plants certainly can take cooler temperatures than the Amazonica ones, and the hybrid picks up that same trait as the parent does. Okay, so having said that, I wanted to just go back to another video if we can, Danny. And this is our Victoria pollination video, which kind of goes through the process we just talked about in the PowerPoint, but actually seeing it happen right while we're doing it. So this is about an 8-minute video, and we'll join you back here when it's done to have any questions and answers. Thank you. Okay, everybody, welcome back. Now that we have seen the pollination of these flowers in a PowerPoint, I thought it would be interesting for you to actually see us actually doing it in person. We have cut some flowers and actually brought them into a studio format so that it'll be a little easier for you to actually see what's actually happening. If we start over here on the left, you'll see that we've covered a first night flower of Victoria last night and still have the rubber bands on it, but the calyxes are starting to peel away. So that would actually open up today and be a perfect flower to pollinate. In the middle is one that was actually wide open and ready to go. This is the one that we will actually be pollinating. In the middle here is the second night flower. You'll notice how the color dramatically changes from the first night to the second night. What happens with these is we leave the cover on the flower that we think is going to be a second night flower, and then that morning we come in to do the pollination, we will uncover it, and this is what you'll get right here. Now I've taken out all of the petals on the inside that you can actually see the middle of this flower and where the anthers are, which we'll be going in shortly to collect the pollen. What I thought would be interesting is take a look at the pollinating tools that we use, and they're very primitive. A pair of scissors, a pair of tweezers, and a paintbrush.
We have rubber bands here and a little vial to collect the pollen with as we go. So the very first step in doing any of these pollinations is to uncover the flowers that you want to use. So in this case, we've uncovered a second night flower which shows its kind of pinky maroon color. And like I said, I've already gone in and taken out the petals. But what I'm going to do now is I'm going to cut these anthers which are holding the pollen, and then we'll come in and grab those anthers loaded with pollen, come over here, and we'll take a look at the first night flower. So let me begin by just taking off some of these anthers, and once I've collected them, we'll be back with you. The yellow that you're seeing, powdery yellow substance in the middle of this flower, is all of the pollen that's being shed. So this plant is very copious with pollen. It is in perfect shape to be pollinated. So I'm going to, now that I've exposed the pollen, I'm going to cut some. And then we will follow back up with a pair of tweezers and the vial to collect it. And then we will head to the first night flower to actually begin the process of prepping that flower. So a simple vial, it can be glass or plastic, will work. And all we're trying to do is get the pollen. We're not removing the pollen from the anthers. I'm taking the entire anther with me, and we will drop that down into the flower. Okay, now that we have collected the pollen from a second night pink flower, put it into this vial, we'll take this to our first night flower right here, and we'll begin the process of preparing this flower. So before we can do anything, we need to go in and remove some petals. So I'm going to come in here and take off some petals to expose the stigmatic surface. The other thing that is important at this stage is that we don't want to contaminate this flower with its own pollen. So we are going to emasculate this flower, which basically means removing all of the anthers so there's no chance of any contamination happening. So I'm not sure, hopefully you can see this, but there is a ring that develops down here, and my scissors are intentionally curved so that I can literally push my scissors in right below the entire ring of anthers. And if all goes well, this will come out in one giant ring that will ensure us that there are no pollen left in this flower to contaminate this flower. So little curved scissors in underneath, poke it through the flower and actually cut it. Poke it through the flower and actually cut it. Poke it through the flower and actually cut it. And go all the way around the entire flower. And when I'm done, you will see the actual ring will come out in one solid piece. So now that we have removed the entire ring of anthers, there's no chance of any contamination. That also opens up this bowl-shaped area inside of here, which is critical for distributing the pollen. This is the stigmatic surface, so this is where the pollen will adhere to, and also where the beetles would have been if they were actually down in there pollinating. So they entered this flower through a hole, walking around in there, spreading the pollen around. To imitate that action, the pollen that we just collected is going to be dropped into here, and then with a paintbrush, I'm going to move it around on the bottom of the surface. So let's go ahead and actually take the pollen that we collected, drop it into the flower, and then with a paintbrush, very soft bristle paintbrush, we're going to imitate beetle feet. We're going to be walking around on the bottom of the flower. So you literally, these anthers which are shedding pollen are laying in the bottom of this bowl, and I am literally just kind of tweaking it around the bottom, just trying to cover the entire bottom surface of this flower with the pollen grains, which will then germinate and unite with the eggs that are right below this. Now that we've actually done the pollination, the next step is to cover this flower back up. This is a little bit tricky, but there are 4 of these calyxes on here, and I'm going to literally tie them back together with a wire. And what that'll do is it'll protect the flower from rain or from any kind of wind getting in there and blowing things around. So if I pierce this calyx, which is fairly thick and succulent, and I come across and I hit the other one, I can pull this together in such a way that it will cover itself up. And then I'll do the same thing with the other two, which are at right angles to it. So if I pierce it here, pierce it here, and then slowly bring it across. And that just gives a little bit of protection to the flower itself. And then sometimes I end up with these dog ears kind of laying out. I just remove them because they're not really doing anything at all for that flower. And if I pull this out of your way, this is the kind of the end result of a pollination that's been done and recaptured. The last thing to do would be to put one of these bags back on the flower so that we can cover this back up and allow the pollen grains to germinate without having anything get in there and contaminate it. So we've just finished pollinating and now we've covered the flower back up. The last step, and probably one of the most important for us, is actually labeling the flower. So the label on here you can see is actually creating a Longwood hybrid crossed with a Longwood hybrid, and today's date's on here. And as I turn this around, you're going to see that right below where we pollinated, this is the ovary area. And this is the area that if this was a successful pollination, that will swell to be about the size of a small cantaloupe. So simply taking this tag, I can take it and just wrap it around here. And this ensures that we know what cross we did, because remember, we're making 3 different crosses. We have to get seed of the parents, both Victoria cruziana and Victoria amazonica, as well as the Longwood hybrid, which is a cross between them. So I hope this was helpful to you guys, and enjoy the rest of your summer.
Okay, well, welcome back, everybody, and Justin, I see you up there.
Hey.
I'm not sure, if there's any questions that we can field, or I know that we're going to be answering questions throughout the time while this video is up on the website. But if there's anybody who has any questions now, I think we have a few minutes to answer questions.
Yeah, we have a few. There's actually a lot of questions, and so we probably can't get to them all, but I would like to throw a couple out there. And before I do that, I'd just like to say my paper that I'm taking notes on is just full of scribbles. That was, I've seen you do this a couple times, but every time I see your presentation, I learn more. So that was fantastic. A couple of the questions, a couple questions here that we had that I thought were sort of pertinent. One was Victoria seed can be difficult to germinate. They would like to know what your germination rate is for the 3 varieties. You had mentioned that Amazonica was a little bit low. And they asked, do you have any tips for a hobbyist to increase their germination rate?
So, I mean, we'll start with the easy one. The Longwood hybrids seems to be, for us, we get probably 90 to 100% germination. Cruziana, we can get anywhere from 60 to 70, but it is Amazonica that is the problem for most people. And that is certainly one of the goals going forward with the Society and with the Victoria Conservancy, is that we offer seeds of all 3 of these, but inevitably, I don't know that anybody's getting germination of the Amazonica seed. And one of those reasons might be the way we store the seeds. One of them might be the conditions we use when we ship the seeds. But we will be working on trying to understand that a little bit more with some research that's coming up, hopefully, in this next season. To see, is 55 degrees a little too cool to store Amazonica seed for more than a year? Because anything that's over a year old, we get nothing to germinate. We only get germination from the seed that we previously did the previous year. So it's only been sitting in the cooler for maybe 6 months at the most. So that's certainly in the pipeline to try to understand a little bit more about Amazonica and what it needs to be successful. And it is an important part, because without it, you really can't create the hybrid. As far as temperatures go, you typically germinate these things in heated water, which can be anywhere from 72 degrees for Cruziana to 80 for Amazonica. And then the hybrid germinates in that whole range. It doesn't really seem to matter. So definitely warm water, more to come on Amazonica.
And there has been some talk about nicking the operculum to help germination. Is that something that you recommend, or do you guys do that there?
Yeah, so that was some research that came out from Kit Knotz's work down in Florida. I think we haven't had to, because I think our post-harvesting process of putting them in the layers of sand, the abrasiveness of the sand, I think actually takes care of that for us. I've actually looked for the hypericums and I couldn't even find them. But there is a little flap that sits right over where the cotyledons, I'm guessing that's the right term, would have come out, that if it's removed, seems to aid in germination.
Okay. The subject of fertilizer is coming up for everybody's presentation. It's something that's been asked a lot on Facebook and seems to be a topic of great interest. Would you mind telling us a little bit about your fertilization program at Longwood, the type, the NPK ratios, and the frequency?
Yeah. So for Victoria, we use a two-stage process for fertilizing, and when I tell you this, everybody I talk to does it a little bit differently. They're definitely heavy feeders. I mean, just to put that right out in the front. It's really difficult to overfeed them once you get them established. Sometimes they can be a little tricky to fertilize in the beginning. So let's talk about, like, in the beginning, what we do. So we mix fertilizer into our soil when we're transplanting the seedlings from that community pot. First, in the community pot, there's no fertilizer. We sow the seeds in straight soil, let them germinate. When we prick them out and move them into individual pots, that's when we start fertilizing them. And we're using pond tabs that we crush up. And to give you an idea of how many pond tabs we're using, we're probably putting, in the very beginning stages, maybe 15 pond tabs in 5 gallons of soil that are crushed up, mixed together very thoroughly, and transplanted. Then the next time we make that transplant, we might go to 20 pond tabs, and then kind of keep increasing it as we go, as the plants get a better root system, and it kind of tends to help keep them from burning any seedlings, because that can be an issue early on, is seedlings get burnt by having too heavy a fertilizer. Now, when we switch to outdoors, we switch over to Landon's fertilizer. So Ken Landon, who we talked about, developed that fertilizer. In the beginning, people were having problems with it being a little bit too strong. But once a plant is established, we find that the plants can take all that food that Ken's developed in that fertilizer and utilize it. So we mix 5 ounces of fertilizer, Ken Landon's fertilizer, with 5 gallons of soil. So we literally take a 5-gallon bucket of soil, dump it into the pots that we're going to pot the Victorias into, mix in 5 ounces of fertilizer, and repeat that process until the pot's filled with soil. That gets us going for about a month into the season. And then starting in July, we'll start supplemental feeding. Our pots that grow the Longwood hybrid water platters are 6 feet in diameter. So they're huge pots. They don't move. We'll put 30 or 40 pond tabs in there every week. We kind of divide those pond tabs up into quarters, and we push them into the soil in kind of 2s at a time. And then we'll also make what are called burrito bombs. So these are basically Ken Landon's fertilizer wrapped up in paper towels that are shoved down into the soil as well. So we have a kind of and that's done about every 3 or 4 weeks.
Wow. Yeah, that is quite a bit of fertilizer. When you say heavy feeders, you really mean it.
Yeah, I don't think you can give them enough fertilizer. I mean, we've been talking about that this week. The more you feed them, the more they grow, and just think about it. I mean, those leaves they're all leaves. So
yeah. There was a question that came in that I hadn't even thought of, but I think it's pretty interesting. You had mentioned that the flowers are exothermic and that they're producing a lot of heat, and you said 10 degrees warmer than the ambient temperature, which is interesting. The question is, do we know the process that produces the heat in the flower?
Well, I mean, it's not an uncommon process. I think lotus do the same thing. I always call it thermogenesis. I mean, if you think of like skunk cabbage is a really good example of that. Anything that's in that aroid family would do this. And, its purpose is probably multifolded, from attracting insects to germinating of pollen grains. But The area where we found the most heat when we did some tests with thermometers, we actually stuck thermocouples into the side of the flower, right in that area where we cut the ring off. And that's where we took the temperature, kind of right in the middle of that cavity. And I don't really know what generates that heat. I don't know, is it the starchy appendages that are generating that heat? I haven't been able to figure out where the heat's generated from, but we do know that, because it's all closed up, the heat builds up in that flower, and it does aid in getting the beetles to the flower, as well as we think probably aids in the germination of the pollen grains. But I really don't know what makes the heat.
Yeah. We're getting close to time, but there are a few more questions if you have a second.
Sure.
You'll often see pictures of babies and people standing on Victoria leaves. What is it that makes them so buoyant?
Well, I mean, there's a specialized tissue called a rhynchoma tissue, which is very prominent in a lot of aquatic plants, and it's basically a term that's used to describe spongy tissue. I mean, there's numerous canals and air pockets that are formed, but on Victoria, as that leaf unrolls, if you were to turn that leaf back over, there are air pockets that can be 2 or 3 inches deep underneath of this, that are created by this kind of network of veins and membranes that kind of create that pattern on the bottom side of the leaf. If anybody, if you haven't seen that, you really need to just go online. You can see pictures of it, I'm sure. But there is air that's trapped in there. There are the tissue itself is filled with these just vessels that are in this kind of a rhynchoma state where it's just spongy. They say that a 6 foot plus leaf can hold 125 plus pounds, which immediately gets little kids thinking they're going to start stepping on them, and then that quickly turns into a little kid going through the leaf. And so that kind of buoyancy is kind of tied into a bunch of things, but those cavities certainly help, and then the actual tissue, the makeup of the tissue plays a role in that as well. Right, what we did find is right in the middle of the leaf, that's where the deepest cavities are. So that's where the most capacity for weight is. As you move out to the edge, the membranes just kind of taper down to almost nothing, where the lip on the leaf is. So that's interesting to kind of look at how that works. And so distribution is always important whenever you're trying to maximize the weight capacity. And you don't see people standing on them without some kind of support.
Yeah.
I mean, little kids were sitting on the middles of leaves in some calendars many years ago I saw, but.
And so the upturned leaves of especially Cruziana, that's pretty unique to Victoria in the water lily world. Are there theories as to what benefits that has?
Oh, absolutely. I mean, there are 2 things that happen with that lip. One is it's also very flexible. I mean, you can tap it and it moves back and forth. There's not much rigidity to that lip. It forms on its own as the leaf develops. But that when wind hits that leaf, it actually moves those leaves around. And we didn't talk about the spines or the prickles that are on the leaf itself, but they are loaded with prickles on every part except that upper surface. As this leaf moves around, anything that gets in that plant's way is going to get scarified. Whether it be water hyacinths, water lilies, even its own leaves. I mean, if you've been on collecting trips, these plants are ratty looking because the new leaves are constantly running out and falling on top of the older leaves. They get scarified up, decompose, fall down into the water, and so it makes room for itself that way. Now, the other thing that I kind of think that they play a role in, that this lip, one of the things that these plants are most susceptible to is wind damage. If you get winds, if these leaves get flipped, they don't flip themselves back over, and usually where they flip, they kind of crease, and that'll quickly decompose. So these lips, especially on Cruziana, they blow in and they put downward pressure on that leaf, and I think they help keep the plant on the water surface during heavier winds.
Oh,
so
interesting.
Besides moving them around and making room for themselves, I think they also aid in preventing wind damage.
Interesting. Just one more question, and before that, we had a comment come in. I think it was in reference to those prickles. He said his fingers must be made of iron by now. Mine are bleeding just even looking at him touch those flowers like that.
I think you just get used to it. And you know how to handle them. I mean, it is 30 years of getting stuck. I still get stuck.
Yeah.
Yeah.
All right, last question, and then we'll wrap it up. Is there Florian wants to know if there's some sort of recording system you have for your tags so that you're not constantly just monitoring the tags that you have in the ponds.
So yeah, well, that tag stays with the flower so that when we harvest the seed, we know what it is. But I take that same information and I put it into a simple Excel sheet. And so, because that's really important, because as you get 10 or 12 crosses going, you can't remember how many hybrids you made, how many Cruzianas you made, how many Amazonicas. And for me, I have to know that I have, several of each going. So I just simply chart it out in an Excel sheet, so that I know what I have. And then I can look at that sheet, which tells me which pool it's in and what the cross was.
Terrific. Well, Tim, thank you for the presentation. Thank you for the time answering questions. Really learned a lot and really appreciate it.
Well, thank you all. I mean, it's going to be fun over the next few years because we do have a lot more work to do with Victoria, and hopefully we'll learn a few more things as we move along and enjoy the rest of the symposium. Absolutely. Thank you so much, Tim. Take care. Okay, so for you attendees, before you take a break here, Danny Cox is one of our computer wizards from behind the scenes, and he has a quick message for you.
Hi, everybody. So you may have been seeing a notification that the new sessions have started as we've been kind of getting towards the end of the current sessions. At the end of each session, if you could leave the session that you're in and move to the next one. That way, when you post your Q&As and your chats, we're able to keep track and monitor them moving forward. If you're in a previous session still and streaming, we may not get to see the Q&A and chat that you have because it'll actually go to the previous session time, not the current session time. So we just finished Tim's session, and now we're going to get ready to jump into Rob's session. So if you're streaming on Huva, which you should be, please leave this session that we're on now and join Rob's. We'll be here. We're not going to start without you. We're going to wait about 2 minutes just so everybody can make that transition, and then we'll get started with Rob's presentation.
All right. Thank you, Danny. I'll be doing the same thing. So we'll see you guys in a couple minutes, and we'll start Rob's presentation.